It's not even funny. Here we are again. So we eat lots of ATP, right? So aerobic respiration is generally what we're going to do. Like I said, the anaerobic is really just reserved. And often for us, it's just something that would occur in a very active muscle, right? So we're going to move that pyruvic acid on into the mitochondria, and everything else is going to take place in mitochondria, right? Eventually, we're going to get all these coenzymes oxidized, but we haven't gotten there yet. So let's start with this. Pyruvate enters Krebs cycle, right? And it does this after it's converted to acetyl groups. And in the acetyl conversion, we do see that the pyruvate to acetyl does actually produce some more NADH, right? And what we're going to do, we're going to release energy from the pyruvate as electrons, right? So pyruvate becomes oxidized, so it loses electrons, loses hydrogens. These are donated to NAD and FAD, which of course become NADH and FADH2. And I said even there's a little bit of guanosine, so but you don't have to worry about the GDP. So they are reduced, right? The carbons and oxygens in pyruvate are used to produce carbon dioxide. So everybody's got a fate. They got an outcome, right? Hydrogens, electrons go here. The carbons and oxygens go here. Oops. All right. So I say the energy in these electrons will be used to make lots of ATP. Low energy electrons will be given to oxygen, and water will be the final product. All right. So we're going to go from this set of reactions that we call Krebs cycle or citric acid cycle to this set of reactions in which is the electron transport chain, and NAD and FAD, the coenzymes, are oxidized, and as they're oxidized they release electrons, and the energy in electrons is donated to ATP. So energy will be used to make lots of ATP, and then the low energy electrons are basically just moved to another molecule, which is oxygen. Again, the reason we actually breathe. All right. So here, that was the outline of that. Here's kind of looking at the transformation of pyruvate to acetyl-CoA, and then what we call the Krebs or citric acid cycle. You, of course, do not need to know the details on this. You do need to understand that all of this happens in the mitochondria. It begins with 2 pyruvates because 1 glucose was catabolized to 2 pyruvate molecules in glycolysis. We're going to end with 8 NADH and 2 FADH2. They will continue on, and then 2 ATP are made and can be used, and 6 carbon dioxide are produced through these reactions. And this carbon dioxide is what you exhale on a regular basis. So this is why you have to constantly exhale. It's because you're always producing CO2 as you use oxygen. So here, again, this is showing the carbon dioxide, right, coming off of the pyruvate. We produce this acetyl group. The coenzyme A is added to this, and in the process we get an oxidation of this and a reduction here. And so the end transformation is acetyl-CoA, all right, with some NADH. In fact, 2 would be made here because 2 pyruvates would do this. So 1 here, and we'd run it a second time and get 2. All right, then this acetyl-CoA comes in, all right, shown here, and it combines with this other molecule called oxaloacetic acid or oxaloacetate, which has 4 carbons. The coenzyme part leaves because it was just acting as a shuttle, right? And we get citrate. So citrate starts this off. It is converted because each arrow again is enzymes and reactions. Converted to an isocitrate, which is, again, you don't have to know that, but understand then that an NAD is reduced, and we lose a carbon dioxide here as we go to ketoglutarate. As ketoglutarate becomes succinyl or succinate, we get another reduction of NAD and another loss of carbon dioxide. We get a formation of GTP, which then allows us to make ATP from it. we do another, right, set of oxidations and reductions here from succinate to fumarate. And then lastly, when we go from malate to oxaloacetic acid or oxaloacetate, we do the last reduction of NAD. And so in all, you get 8 of these. There's one, right? Excuse me. 1, 2, 3 here, but we do this twice, so 6, and you had 2 up here. You have 1 FAD here, but we do this twice, so you end up with 2 FAD and 2 ATP. All right, so that's still not enough energy for us to live, so we go into the electron transport chain, which is a process of oxidative phosphorylation, and begins with these NADHs and FADH2. These are the reactants. They come in, they are automatically oxidized, and they go right back out and start shuttling more. All right, so they start this whole chain going. The hydrogens and electrons are donated here, so these two things are oxidized, and they return, and energy is captured, right, by shuttling all these hydrogens back into the matrix, because they get pushed out through this big membrane complex called ATP synthase. And that's what it does. It synthesizes ATP. So electrons are transferred to ATP, and they're showing that happening here. And these spent electrons, very low energy electrons and hydrogens, are given to oxygen to make water. All right, so just how much can be made? We just kind of walk through this idea of, well, here were 2 here, there were 2 here, and then to make the rest, so there was only 2 places before the electron transport chain that we saw ATP made, and it wasn't much. The majority of ATP made really comes from, right, energy carried in these 2 coenzymes. So as they come in, right, and roughly there's about 3 ATP that can be made for every NAD, so that's where you get 30 ATP, and there's 2 ATP that can be made for every FAD, so we get 4. So if I can actually, right, oxidize the NADH and FADH2 completely, I could make 34 ATP in the electron transport chain. So payoff is the electron transport chain because of this oxidation, and this kind of walks you through then the math there. All right, if glucose is low, what do you do, right? So remember, we're using glucose to do all this, so what happens if I can't start the whole reaction? Well, I die, so I have to have other ways, right, to start this reaction. So we talked about this process back when we talked about hormones that we call gluconeogenesis, so formation of new glucose. So you can make glucose. Yay, right? That's really good news, right? So this would be done again in that famine state. You can start with pyruvate, you can start with lactate, glycerol, alanine, or glutamine. Any of those can be used, and these are amino acids. This, of course, came from fatty acids, from triglycerides, separate from the fatty acids. And this is if we got to, right, either one of these carbohydrates. Mostly done by the liver, because the liver has, right, it makes a lot of choices for us in metabolism, and liver has all the enzymes to do this. So if you're fasting, starving, or on a low carbohydrate diet, right, then you need to make you need to make new glucose in this process we call gluconeogenesis. And I say, why? Well, because the brain can only use glucose for energy. I say this again several times. I've said it already. Please don't forget it. And we have a constant we keep in our plasma, 70 to 110. So this kind of walks you through the actual biochemistry, which is good to be aware of, but you're not going to draw this out for me on a test. Okay, this is really capturing what you need to know. All right, so for lipids, lipid metabolism. So we've covered everything we do with carbohydrates. What about these lipids that you've consumed in your diet? Just doing your job, right? So again, we had these triglycerides are common. So a glycerol with 3 fatty acids. Two of the fatty acids generally are freed, right, by breaking bonds here. and here, and then you produce a monoglyceride with 1. So this is basically free fatty acids and monoglycerides is how you would absorb, and these absorb by diffusion, right, because they're nonpolar. So I say you either ate these or the adipocytes and hepatocytes made them. And I said most likely you're doing both, right? So you have consumed them and you've got them to this point through digestion, or your fat cells and liver cells are basically making them because you need fats, right? Fats are very important to us. This is a lipoprotein in a cartoon here. So it shows you, again, the creamy center. I kind of liken these to Oreos, right? The outer. I mean, in Oreos we use to demonstrate the membrane, but it's a very good demonstration because the outer part dissolves in milk and the inner part doesn't. So you got your polar to nonpolar. Well, that's the way you transport all these triglycerides in the blood because blood is water-based. And so the outer part is a lot of the polar, including embedded proteins that we call apolipoproteins because lipid proteins, right? But they're proteins. And then the inner is this creamy nonpolar center that stays away from the blood and all huddles together, all right? So when you ate them, most of the digestion occurred in the small intestine through lipases assisted by bile, and they were absorbed through simple diffusion, as we talked about in that chapter. Once made into chylomicrons, and this happens in those absorptive cells of the small intestine, they then enter. And so this is a typical cartoon. Again, chylomicrons are a type of lipoprotein, right? They are transported in the circulatory system this way so that they can stay in solution. These are lipoproteins, and why do we need them? Well, they are delivering this and allowing, right, lipids or nonpolar stuff to stay among polar molecules, blood. All right, what can we do now? Well, several options. We can break them down, called lipolysis, and this would be very advisable if you were in famine state, right, low energy. Splitting the triglyceride into fatty acids and glycerol, which have different fates through lipoprotein lipase, and this is inside cells would contain this specific enzyme. All right. Fatty acids take part in beta oxidation, so you're breaking them down to acetyl-CoAs, and once I get to an acetyl group, I can use them in the mitochondria of any cell to make ATP, right, as we did with our glucose in cell respiration. Glycerol, the other part of the triglyceride, can either enter glycolysis or can be used for synthesizing glucose, so gluconeogenesis. So again, here's a nice cartoon to kind of walk you through what happens to these, but this is really the important part. What do we do with each of these? All right, ketogenesis, this comes up a lot in people's minds, especially with the fad diets, ketogenic diets, all right? And so what exactly are ketones? How does this work? So here's what it is. So if there's excessive acetyl-CoA, so this is made again through the process of beta oxidation, breaking down fatty acids, all right? And there is a diversion into creating ketone bodies, and this is done a lot by the liver. And so the liver takes this excess, right, because you get a lot of energy in fats. These excess acetyl groups, as we're seeing here, all right? And they're converted, in the arrows again represent the biochemical reactions, through this pathway, right, to acetoacetate, beta hydroxybutyrate. Right? These are basically ketone bodies. So ketone bodies are energy-containing molecules when glucose is low or unavailable, so like in a diabetic. And if they are made in excess, then we talk about this, right, ketosis or metabolic acidosis, or ketosis specifically, all right? So the liver is very good at doing these pathways because the enzymes it has, and it just starts dumping these into the blood, all right? So these ketone bodies can be oxidized then, right, to release energy. That's the whole point. So we have an oxidation here of beta hydroxybutyrate to acetoacetate to acetyl-CoAs, which now I can enter into Krebs. So I said, so you are getting that ketone. So hopefully you're getting the ketone bodies are alternative fuel, right? And if glucose is low, your organs can use something else to get ATP. And they say here, if they're produced faster than used, you get acetone and carbon dioxide. Acetone is exhaled, and the CO2 acidifies blood.